Voilà, je vais faire la présentation en français. Merci à toutes et tous d'être venus. Aujourd'hui, on a avec nous la docteure Jessica Allen. Elle est professeure adjointe à l'Université de Eastern Washington depuis 2018. Elle a réalisé son PhD in the New York Botanical Garden et puis elle a poursuivi sa carrière à, je vais le dire, Swiss Federal Institute of Forest, Snow and Landscape Research. Donc, sa recherche se concentre sur la génomique, l'évolution, la biodiversité aussi et la conservation du lichen. Elle a récemment publié un guide du terrain sur les lichens trouvés du nord-est de l'Amérique du Nord. En plus, elle a de nombreux articles dans des revues prestigieuses. Aujourd'hui, si je ne me trompe pas, elle va nous parler de l'espèce de lichen menacée, Cetradonia linearis. Plus particulièrement, elle va discuter la génomique populationnelle de cette espèce et un possible lien avec la communauté microbienne y associée. C'est un sujet passionnant. Au labo, on connaît bien ton travail, on l'aime beaucoup. Et donc, juste remercie d'être venu. Merci beaucoup. Thank you very much for being here. And you can start.
Great, thank you so much for the invitation. I'm really looking forward to sharing some of this research with you all and to hearing if you have any insight into some of these results. Some of them are preliminary and some of these analyses are ongoing. Can you all see my screen okay?
Yes.
Okay, excellent. So I'll be talking about the rock gnome lichen today. So the green thing underneath this very cute salamander. But it's nice to put an animal picture at the beginning of a talk sometimes to draw people in. Okay, so I'll start with a quick introduction to lichens, which, I do find that Canadian citizens tend to be much more lichen aware than in some countries. And of course, you have some lichenologists in your midst, so you're probably already up to speed with the lichens, but just in case anybody's tuning in and hasn't been introduced to these wonderful organisms, I'll just start with some basics here. So lichens, first of all, are beautiful and diverse. To me, they're really the quintessential symbiosis, and they occur on every single continent on this planet. There are over 20,000 described species of lichens. Their names apply to the fungal symbiont, the main fungal symbiont, and up to 20% of all fungi are lichenized fungi. That is, they form these lovely symbiotic structures. And these lichens perform numerous essential ecosystem services, providing food and shelter for animals, both invertebrates and vertebrates. They're really important in nutrient cycling and other processes like soil formation and soil stabilization. They're also very well known as bioindicators, both of air quality and sort of detectors of other pollutants like heavy metal pollution. Now, people are usually really familiar with our sort of textbook definition of what lichens are. These symbiotic organisms formed by mutualistic interactions between a fungus and an alga and or a cyanobacterium. But over the past couple of decades, it's become increasingly clear that lichens are much more than that. And indeed, if we're to take a closer look at the lichen body and try to deconstruct it a bit, and we call that lichen body the thallus, we find that there's actually a fabulous wealth of diversity in every single lichen, right? So we have that main fungal partner, that dominant fungus that we usually call the microbiome in this relationship. This really provides most of the scaffold for that lichen structure, most of the biomass. Those photosynthetic mutualists, which we usually call photobionts, are green algae and or cyanobacteria, and and it turns out that usually there's actually a community of these photosynthesizing partners in there, even if there might be one dominant photobiont. Now it also turns out that there are a lot of other organisms in these lichen structures. So there's usually a really diverse endolichenic lichen or fungal community. So other fungi involved in this structure, these tend to form either commensalist or parasitic symbiotic relationships. There's a wealth of endolichenic bacteria, right? These communities are incredibly diverse, serving you know, fitting many different roles in these lichens. There are also many microinvertebrates, especially nematodes, tardigrades in and on those lichens. And then I also like to point out that the chemistry of lichens is really unique. So these structures produce many, you know, many hundreds of different secondary metabolites, most of which are not observed in other groups of organisms. And those chemical compounds serve many functions, both in mediating these interactions inside the lichen thallus and also interactions with the ecosystem at large. In short, lichens, while perhaps appearing humble and inconsequential at first glance, are really hubs of diverse interactions. And we're really just starting to understand how each component of the lichen symbiosis acts on its own and in concert with other components of the thallus. And there are so many outstanding questions about lichens that both can give us insight into how these unique organisms work and also how symbioses and biotic interactions work broadly. And I'll touch on just a few of these questions briefly today. So the first, you know, it really focuses on trying to disentangle those interaction networks in this microbial microcosm. You know, my second question is really, how do abiotic factors and biotic interactions influence the different components of the lichen holobiont? And then how does the population genetic structure and the microbial microcosm vary between rare and widespread species? And we'll get to that in a little bit. Okay, so I'll start by talking about the mycobiont, that main fungal symbiont, and then I'll talk about the bacterial communities, spend a bit of time, just very briefly start to pull these two together, think about how they might be interacting. So to really highlight these different components of the lichen symbiosis, I'll start by discussing the population genomics of Cetradonia linearis, and then looking at the bacterial microbiome of that same species. And then finally, getting back to that main fungal symbiont, I'll share a little bit about some of the lichen genome sequencing we do in my lab, and a little bit about tying that into undergraduate education and genomics, in case there are any educators here in the audience. Okay. So the rock gnome lichen, right? So Cetradonia linearis, this is one of the earliest diverging lineages in the family Cladoniaceae. It's narrowly endemic to the southern Appalachians of eastern North America. So this is its entire global distribution. Its closest relatives are all found in Australasia. So this species is likely a paleo-endemic of this region. It is one of only 2 lichens that's actually protected by the Endangered Species Act in the United States, actually one of 2 fungi at all. It is listed as endangered and has been listed as endangered since 1995. We assessed it against the red list criterion, and it's now on the IUCN red list, the global red list, and we assessed it as vulnerable globally. There are about 100 known populations and maybe 4,000 mature individuals in existence. So quite rare and quite restricted. It has very specific habitat requirements. So it only grows on rock, and it can be found on high elevation rock outcrops, especially at the base and especially in shaded areas where there's some seeping water. And it forms sometimes some fairly large patches on these types of structures. Incidentally, sampling these sites tends to be really fun. So here's my field assistant actually rappelling to get to one of them. And it also grows on large boulders in streams, in sort of low to mid flow streams. Again, these streams tend to be at higher elevations, and it especially grows next to waterfalls quite a bit. Again, sampling for this lichen was an absolute dream. But in short, it really prefers areas with like constant moisture and cool air flows at these really mid and usually higher elevations. Here's a bit of a closer look at it. So It's composed of these sort of finger-like projections that we call squamules, and the upper surface is a light green and the lower surface of these are white, and they form these really discrete patches. And they often do produce reproductive structures, so you can frequently see them with apothecia. These are structures that form the fungal sexual spores, and also with pycnidia, which form conidia that are the asexual spores or act as spermatia. And you can see those, they grow at the sort of ends of these sort of somewhat finger-like projections, as I like to call them. Okay, so because this species has been listed as a federally endangered species for quite a while, there's been, over the decades, quite an effort to very carefully map the distribution of the species, its population size, and a lot about its ecology. However, when I started this project, there was no understanding of its sort of population genetic structure, its genetic diversity, anything like that. So I came at this from a conservation perspective, trying to understand how genetically diverse the species is, what is going on with its reproductive biology as far as sexual versus asexual reproduction goes, and really how connected are these populations, because the sites where this species occurs tend to be fairly isolated from each other across the landscape. And I had 4 hypotheses going into this. I expected to find that most of its reproduction and dispersal would be clonal. I was really envisioning those structures breaking off from rocks, especially in streams, maybe floating downstream, getting stuck on the next rock. We have a fair bit of clonal reproduction going on. And especially because I saw conidia more frequently than I saw apothecia. I did expect to find a strong geographic structure to the populations because the species distribution is so patchy across even the small landscape, and because I really went to this with the assumption that indeed it is a paleo-endemic and that its distribution was really strongly shaped by Pleistocene glaciation. So the Southern Appalachians were never glaciated. And I suspected that if it was actually good at dispersing, that it would have ended up moving up into the Central Appalachians as the glaciers retreated, and we don't see that happened. I did not expect to find evidence for adaptation to different habitats, mostly because the niche of this species is so narrow. When you look at the climatic variability that it can sort of withstand, there isn't a great deal of variation there. And then thinking again about the biogeography of this region and of this species, I expected to find some evidence that it was potentially more widespread during the last glacial maximum. Okay, so to get at these questions and hypotheses, I collected samples throughout the range of the species. So its total distribution is shown with the black dots. The white dots are populations that I sampled. At each site, I collected 2 or 3 individuals with sterile forceps directly into sterile tubes. And this was, in the end, I was working with 32 individuals. Again, this is a federally endangered species, so very limited sample size, but I did what I could with what I had. And then I conducted whole genome shotgun sequencing of all 32 of those individuals, assembled the genome from those data, and then called the SNPs from those data with those 32 individuals. Now, the coverage of the sequencing was enough to put together about 50 to 60% of the genome. So we ended up with a 19.5 megabase assembly, recovering about 6,000 genes there. And then the SNP set was comprised of about 10,000 SNPs, which is what we landed at after correcting for linkage disequilibrium, so removing linked SNPs from the data set. Before proceeding with any other analyses, I checked for clones in this data set using a cutoff of 95% identity. And then most of these analyses were conducted with the R package popper. Okay, so first I built a neighbor joining tree, and what the result of this was that all of the individuals sampled from the same site clustered most closely, and all of the sites that were in the same vicinity of each other, the same mountain range, clustered together. So already seeing some geographic influence there. I then ran instruct to investigate some of the population structure here, some of the clustering of these individuals. And in this situation, I ended up with essentially four gene pools, and we can see here the proportion, the sort of inferred proportion belongingness to each of these different gene pools, and that each individual has sort of a dominant gene pool. And then looking at this in a geographic context, again, we can see that sites that are closer together are sort of have similar composition, similar sort of proportional composition when we look at those different gene pools. So these are the Great Smoky Mountains National Park over here, dominated by that teal. These are what I call the southern Nantahalas, which tend to have that dark blue. These are the Black Mountains with the purple, and then these are the Balsam Mountains, and then this is Roan Mountain that are sort of orange dominant. And I'll come back. This was really a strange thing, but I'll come back to this a little bit later because it's actually that connection between the Balsam Mountains and Roan Mountain has been inferred in other groups of organisms as well. So I ran a Mantel test just looking at the relationship between pairwise FST and Euclidean distance, found a strong correlation there, strong significant correlation supporting that isolation by distance. Okay, then looking at the mating systems, here I have a brief sketch of a lichen life cycle. So lichens reproduce in many different ways. Sometimes these different modes are occurring at the same time. But really, I was interested in the the reproductive system of the fungus, the sexual reproductive system. So I looked for the mating type loci. I found the MAT2 genes in 14 of 32 samples, did not locate MAT1 genes. So I think it's still up to, we still don't necessarily know exactly what the mating system of the species is. It could be heterothallic or homothallic unisexual. I would like to acknowledge Dick Harris's help in this particular area, trying to actually find sort of incipient apothecia and trichogynes and other sort of structures that would indicate some outcrossing sexual reproduction going on. So in that instruct analysis, I chose that analysis because I could infer the self-fertilization rates simultaneously for each of these groups, and that really that came back with about 60% of the time some sort of self-fertilization potentially happening here. So that's pretty high. And then I also looked at recombination using RPD4 and actually trying to infer the number of recombination events between individuals from different within sites and among all of the samples. And we're looking at sort of recombination like events from 14 or 141 overall across all of these samples. So seems to be some low rates of recombination. Okay, so on to this next question, really trying to piece apart that influence of geographic distance and structuring the populations versus any sort of environmental influence on these populations. We ran a partial Mantel test, and again, I already showed you the result of looking at the relationship between genetic and geographic distance. We had a strong positive correlation that was significant, and there were no significant I didn't find any significant correlation between any of these other variables and genetic distance. I will say that I checked for autocorrelation among these three climactic variables before running this analysis. Okay, and then my final question, looking at the biogeography of the species, really I had this, I'm thinking about the species back through time and maybe what was happening for it during the Pleistocene and what that can tell us about what might happen to it in the future with climate change. So what I did was take all of the known sites and I built a species distribution model using those. I'll go back a slide for a second here. I used these 3 climactic variables: mean temperature of the wettest quarter, of the warmest quarter, and annual precipitation, because these are not autocorrelated. These are from the BioClim data set. And I used MaxEd to build the species distribution model here. So areas in red are inferred to be more suitable for the species, areas in blue are less suitable, and then the areas in gray are inferred to be not suitable. So this is sort of the distribution model for the present. I projected this back in time to the last glacial maximum. And based on this model, it does infer that there may have been more suitable area for this species at that time, a few tens of thousands of years ago. And then what I have overlaid on this is just the pi genetic diversity for each of these regions. And we see that indeed at this point, the genetic diversity in the areas in the more southern part of the species range are more diverse. So that would suggest that potentially the species was more widespread at that time, and that its distribution has contracted to the present. And given the projections for climate change in this region, that they expect to see generally hotter and drier conditions, you know, there's the potential for continued contraction of the species distribution into the future. Okay, so, you know, in summary, taking all of these results together, I didn't find any clones, but I found some evidence for recombination, not a ton of recombination, found really strong isolation by distance. Also found that the species is fairly genetically diverse, and have some evidence for that the species was once more widespread. Okay, now when comparing the results for Cetradonia linearis to other southern Appalachian endemics, we kind of get back to this oddity of looking at Roan Mountain, along with a few other kind of key takeaways. So one is that in this study of Desmognathus wrightii, so a salamander, and this endemic spider to the southern Appalachians, Hypochilus bukkake, both of these studies also found very high genetic diversity compared to other species in these groups of organisms, and a really strong geographic structuring, even over this fairly small spatial scale. And they also tend to find that Roan Mountain, which is really, it's up in this area here, and number 4 on this map, isn't actually sort of grouping in either case with these sites that are more like geographically close to it. Either they're genetically anomalous or grouping with these sites either down in the Smokies or the Balsam Mountains. So just interesting to see that same pattern play out with Cetradonia. And then in taking this and comparing it to some lichen population genetic studies, there are many more that out there, but just trying to think about this in sort of our grand scale of some of these different life histories, different reproductive strategies, ecologies of these organisms. I kind of think of Cetradonia falling in the middle in some ways, especially looking at rates of clonality versus outcrossing. And, you know, with some species like Loberia pulmonaria, where we see high rates of clonality, versus other species that seem to be really admixed over very large spatial scales. Maybe Cetradonia is kind of in the middle there. And then this is the part that I'm really excited about. So I was delighted to see this population genomic study of Cladonia stellaris come out from Marta, Juan Carlos, and have the chance to actually compare it to some of the metrics that I found with Cetradonia, because they're both in the Cladoniaceae, and kind of give me at least something to compare in thinking about this question of looking at really rare species versus really common and widespread species, and how do their population genetics patterns, like how do they actually measure up? And these two species are different in a lot of ways, so it's a bit of a hard comparison to make. Of course, one of them is their total distribution size. So here's the distribution of Cladoniaceae stellaris. These are data directly from the lichen portal that I haven't cleaned or anything like this, but you get a sense of the abundance and how widespread it is versus Cetradonia linearis, which I have as this little yellow dot here. So again, widespread abundant species versus very narrowly endemic. Again, they're different in a lot of ways. The frequency with which they produce any sexual reproductive structure, their habitat preferences. We have a soil-dwelling Cladoniaceae stellaris versus rock-dwelling Cetradonia, and the overall morphology, of course. However, I still think it's at least an interesting thought experiment to put these side by side and really think about the fact that the pairwise FST values in Cladoniaceae stellaris are so much lower, and these are based off of populations sampled on a latitudinal gradient in Quebec versus Cetradonia, which the pairwise FST values are much higher. And this contrasting sort of evidence for some spatial structuring, potentially because of limited distribution, dispersal abilities, I think, of Cetradonia linearis, versus sort of more likely much larger, I'm guessing, with more sampling gene pools potentially for Cladoniaceae stellaris, but looking over a somewhat similar spatial scale. At that scale, we don't see as much spatial structuring with this widespread species. So it seems to be kind of able to get around, my inference is be able to get around better. But of course, I'd be curious to hear your opinions, Marta and Juan Carlos, about this as well. Okay, so next I'll move on to talking a little bit about the bacterial microbiome of this species. So this portion of the study is not published yet. The analyses are still really in progress, but I really wanted to share some of my preliminary results with you all. And before diving into it, I would like to acknowledge that Elizabeth Alter was really the one who sort of instigated us doing this at all. She's worked in microbiomes in a number of different organisms, and really pushed me to include this component of the study. So what we did was essentially take all of those exact same samples that we sequenced the genomes from, did the whole genome shotgun sequencing, and we conducted sort of 16S amplicon based sequencing at the same time to capture that bacterial community. And from there, basically what we've done so far is use DA2 for all of the initial cleaning and filtering, and I've conducted a number of analyses in QIIME2 at this point to address just a few questions here. So I was curious to know if habitat, climate, or geographic distance influence the bacterial community composition in the species, if there's any relationship between these variables and the diversity of the bacterial communities. And then just simply to characterize the components of the communities. And I was really curious about cyanobacteria specifically. So Cetradonia linearis' primary photobiont is a green alga. However, I had some reason to suspect there might be some cyanobacteria involved. Okay, and again, these analyses are really preliminary, and I'm just going to focus on some of the significant results that we've seen thus far. Okay, so first, looking at the relative abundance of the phyla that were present at an abundance of over 1%, what we see when looking at all of the samples is that proteobacteria and acidobacteria are the most abundant phyla. And then after that, the various phyla, they vary by sample. And we can see that in the sunburst plot at the right as well, where phyla are in the very center out to our lowest taxonomic level around the outside edge of this. Okay, and I'll quickly come to this one phylum that I was very curious about, cyanobacteria. And the reason I was curious about this is because Cetradonia in some locations grows like it's almost being overgrown at times by cyanobacterial mats. In some places, the cyanobacteria are very abundant around it. And we cleaned all of these samples thoroughly before doing the 16S sequencing. And I really wanted to know if any of those cyanobacteria were actually like penetrating the thallus and living inside of it, either in the cortex or in more internally. And what I found was that the cyanobacterial content varied quite a bit by sample and by site. And we can look at this in another way, just looking at the relative abundance of different cyanobacterial OTUs. So each column is a cyanobacterial OTU and each row is a different individual. And I have them organized by their habitat, cliff or stream dwelling. And I just wanted to take a closer look at one of these individuals, T2, and take a look again at a sunburst plot just for this individual, because I was really curious, like what cyanobacteria are actually here. And so one interesting result was that Stygonema is one of the most abundant cyanobacterium that we see in this sample. And this is a filamentous cyanobacterium that sometimes actually forms symbioses with lichens. So that was just sort of following up on some of my curiosity there. So to look at the potential geographic structuring of these bacterial communities, I use the Bray-Curtis dissimilarity values along with and I used a Permanova, binning the samples by both sampling site and by mountain range. And I found that those distances, those Bray-Curtis dissimilarity values within groups were significantly less than they were between groups. So we have some suggestion of a really strong location-based influence on these bacterial communities. And these graphs are just showing ordinations and each point represents a sample. And you can see the samples colored by the sampling site on the left and by the mountain range on the right. So some evidence for location-based structure. And then when looking at these different environmental variables, I first looked at whether they were cliff or stream dwelling. And here I did find that sort of using the alpha diversity metric of faith phylogenetic diversity, that cliff dwelling communities were more diverse than stream dwelling communities. And then again, I ran a Permanova on this data set using, again, the Bray-Curtis dissimilarities among between these two habitat categories, and recovered a significant result again. So the communities are different and they're more diverse from the cliff dwelling individuals than the stream dwelling individuals. Okay, and then I looked at that same suite of climactic variables that I used in testing for ecological adaptation for Cetradonia linearis and in the modeling. So I chose these because they're not autocorrelated. Again, among the three of them, they're also not correlated with geographic distance. And only one of them did I end up finding any sort of significant result. So here's a Mantel test between that sort of difference in mean temperature of the warmest quarters versus that Bray-Curtis dissimilarity, and again, strong positive correlation. So, so in thinking about this, thinking about the fact that I guess I was not super surprised that sort of that mean temperature of the warmest part of the year would have some influence on the bacterial community, especially considering that that is the time of the year that I actually sampled these individuals. Okay, so trying to tie all of this together just a little bit. Let me check my time. Yep. So first, thinking about tying together these data for the Cetradonia linearis, that main mycobiont, and comparing it to those bacterial communities and the influences on both of these components of the lichen symbiosis, I did find some spatial structure to the population, to the microbiome populations, as well as the bacterial communities. But in contrast, there seems to be some significant environmental influence on those bacterial communities. While I did not find that same pattern with the particular ecological variables I used in looking at the fungal population genetics. Okay, and then 2021. Anyways, looking at comparing again with Cladonia stellaris, Cladonia linearis, interesting to see that in both of these species, there's some geographic component to structuring those bacterial communities. And in Cladonia stellaris, total domination, it seems, by the proteobacteria, and which was a group that was abundant in Cetradonia linearis, along with many other groups. Okay, so just to give us a little bit of a checkpoint on here where I am with this talk, right? So having covered the population genomics of Cetradonia linearis, the bacterial microbiome, started to think a little bit about bringing these two components together. And then finally, I just wanted to really quickly talk a little bit about some lichen genomic sequencing happening in my lab, and a bit about some undergraduate genomics teaching resources. So over the past 4 years since starting my position at Eastern Washington University, I've been working really in a really focused way on generating very high quality lichen genomes using long read sequencing. And I've been using the Oxford Nanopore platform because I could do all of my sequencing in-house. So I work at a primarily undergraduate institution. And so most of my job is teaching. I also have fairly limited laboratory resources. And this has been great because here it is actually on my desk that I'm sitting at to give you this talk where I sequenced the first genome. That's the picture from that. And just a few key points about our methods here. It takes a lot of DNA going into this process to get a really high quality genome. So I usually do multiple extractions and pool them. Really fine tuning that size selection step has been key to improving our results. So I find that a 0.4 magnetic bead to 1 part sample volume gives me the best results, sort of that point of getting rid of the smaller length DNA molecules, keeping enough of it at the same time. And actually with the new library prep kit LSK 110, throughput has actually become a lot less of a problem. It's been a major upgrade. And we're continually refining our bioinformatics pipelines for the assembly of these reference genomes. And so what I'll show you here is just exactly where we're at right now with this pipeline. And it's shifted quite a bit with the introduction of LSK 110 and some updates in the algorithms that are really key to these analyses. So using the most up-to-date base calling here with the Guppy version 5 and the high accuracy mode with soup mode. Then using Fly for assembly, and what I found with in previous iterations when using LSK 109 library prep kit, I didn't use the meta flag, but what I'm finding now is that using metafly is actually working much better for these assemblies. Some of our samples end up being diploid or triploid, or simply having multiple haplotypes present, especially if I have to pool multiple individuals to get enough DNA. In those cases, there's some extra steps here, essentially removing disjointigs with this purge haplotigs program and then recontigging before moving on to the next step. Polishing that assembly with Medaka, filtering, using homology-based searches in Diamond, and then essentially examining the outcome of that in relation to coverage and GC content and doing some filtering there. And then of course, Busco and Quast looking at some key metrics on the outcomes of these assemblies. Okay, so this graph doesn't include all of our genomes that we've assembled in the lab, but in general, we're finding that our assemblies end up being highly contiguous. So the 5 colored lines show some of the genomes we've put together, and the gray lines show some previously published lichen genomes. So ours are in amongst the best of them. We can see this again with the Busco scores, right? So looking at completeness here, well over 90% for the most part with all of our genomes. And then just in case anybody is out there doing sort of on the browse for lichen genomes, we have a few out, so Latheria columbiana, Latheria lupina, and then more recently Basidia gigantensis, which was a collaborative project with Troy McMullen and Steve Jones that I did all of the analyses for that particular project. So those are all published. They're actually all on RefSeq on NCBI at this point. Lepraria neglecta should be forthcoming soon-ish. And then we have 4 others in the works at this point, including 2 critically endangered species, Cetradonia and Sulcaria icidifera. And then we're sequencing another 12 genomes in the next year as part of my currently funded National Science Foundation grant. Okay, and then my last plug for the day, I did just finish teaching bioinformatics for undergraduate students, and so in the course of 10 weeks, got students up and running with using Linux. We went over bash, awk, grep, and sed using conda. We assembled and annotated genomes and then did some phylogenetics as well. And I did all of this on the Google Cloud platform, so we also don't have a ton of compute resources at my university, and of course students' own computers tend to vary quite a bit. So I moved us all onto the cloud, and so we also learned some cloud computing, taking advantage of Google Cloud's 3-month free trial period. And so I have a whole series of tutorials that I'm just polishing up, and they'll be up on GitHub soon. So if you're interested in that, I'll put my email addresses on the next slide, and you're welcome to email me after this. And once I have that posted, I'm happy to send that out to anybody. And then I just have a picture of my students here doing something maybe slightly more fun than analyzing data. Occasionally we do get to go snowshoeing to collect lichens in the winter. Okay, so a lot of people have been really instrumental in all of this research. I received funding from a whole bunch of different sources to do this work. And I'd really just especially like to thank Elizabeth Alter again, who was instrumental in the population genomics and the microbiome work, along with Jenny Walk, who helped me get up and running with QIIME to get some of these preliminary analyses put together. Okay, happy to take any questions.
Thank you very much, Jessica. It was really amazing. I really like it. I don't know if you have any question